Hey y'all, Dr. G here. This is the last video in our series of videos that talk about nucleic acid structure. We first introduced the base structure of nucleic acids and the idea that they are made up of a central sugar, ribose, and attached to that central ribose is one, two, or three phosphates and a base. That base falls into two categories, purines or pyrimidines. And those bases make important hydrogen bonds that complement one of the other bases. Okay, we talked about how that is important for the overall three-dimensional structure of DNA. It adopts this double helix that is quite famous, that has some very defined characteristics. And if that double helix happens to be RNA or a DNA-RNA hybrid, it has a little bit different structure. In this video, we're going to focus in on the idea of further structures and properties of DNA and RNA molecules in eukaryotes. Okay? So the main difference is that in eukaryotes, the DNA is confined in the nucleus and has a lot more structure to it. And that structure is presented in the form of chromatin. So without getting into too much detail, the idea is that the DNA is wrapped around these histone proteins. And there are 4 histone proteins that are part of this core. Each of these 4 have exactly 2 copies in the histone. Okay, and then DNA will wrap around that histone 2 times. Okay, and that's what we're looking at here. Each of these structures are called nucleosomes. They're linked together by a shorter chunk of DNA that's protected by another histone protein, H1. So kind of collectively, the idea is that they wrap around the histone, and then that wrapping around is locked in place by the histone H1 protein. Okay, and you have a bunch of these beads on a chain that collectively make up the chromatin structure. Okay, you can degrade these, you can do a lot of things to them, but what I really want you to understand is this core structure of DNA in chromatin in eukaryotes. Okay, let's look a little bit more closely at this overall structure. So here what we're looking at is this structure. Okay, so what we can see here in the middle is the core protein structure. And I don't know if you can see it or not, but each of these colors has 2 copies. So the blue here, this is one of the histone proteins, and here is a second copy of it. The yellow, there's 1 copy here, another copy here. Okay, there's 2 copies of each of these, making up a total of an octameric core. And the DNA we can clearly see is wrapping around it. And if we rotate this and look at it from the side on, we can see that the DNA wraps around this octameric histone core 2 times. Okay, so it's wrapping around twice. That's the core nucleosome structure of histones. Okay, there's a lot more to histone structure and to nucleic acid chemistry in eukaryotes, but we're going to kind of leave that for other courses. But for now, what I also want to think about is some important structure for RNA molecules. We often forget that RNA has important structure. RNA, when it's present in a single strand, it doesn't just randomly flop around. Just like proteins, just like DNA, it adopts structure. And the core of this structure is something known as stem loops. Okay, stem loops are, the idea is it's a single-stranded chain of typically RNA that has hydrogen bonds within itself. Okay, so you have, this is the stem stabilized by hydrogen bonds with the bases, and then you have this loop, right? This stem is an RNA double strand at this point, so it is typically A-form RNA helix. Up here, there's not a lot of obvious structure. However, this loop does commonly protect itself, right? Because we don't want to have nucleic acids floating around exposed to enzymes to be able to be degraded. So there are a lot of common strategies that have evolved that allow these structures to stabilize themselves. One really common one is called a tetraloop. In the loop, we usually find a UUCG sequence, okay? And as it turns out, this allows the nucleic acid bases to pi stack along with the A form helix. And on the next slide, we can kind of see that happening. So here we're looking at this UUCG tetraloop. So the blue here shows it, and especially I think in this image right here, you can kind of see how they're pi stacking. They are lined up pi stacking with that A form helical stem, and this one is as well. So 3 of the 4 bases, the first U, and then the C and the G, are able to stack on top of each other, making stable pi stacks. That aggressively stabilizes this interaction. Another really kind of cool feature about stem loops is the abundance of these molecules called microRNAs. Okay, so microRNAs are really important regulatory pieces of RNA. They consist of a really long stem loop, and they can be quite long, but they always have a couple mismatches, as we see here in green. So we have a couple mismatches that are not able to base pair. We have some irregular base pairing here shown in purple. So this adenosine actually makes base pairs with both this G and C through non-canonical, non-Watson and Crick base pairing. And then we also have several non-standard GU base pairs. Okay, so guanine or adenosine and uracil are able, right, so here this GU is able to make hydrogen bonds, make these H bond interactions. They're non-canonical, but they can still happen, okay? So these features are always present in the stems of these microRNAs, okay? So they're usually pretty easy to spot. And the way that they work is you have these microRNAs that get introduced into a cell, okay? And then we have these proteins called exportins that recognize them, and they bind to them, and when they do, they break them apart, they knock off the loop part, they kind of chop off some of the stuff on the 3' and 5' ends, and you're left with a microRNA duplex. This duplex can be split apart by another protein complex called the microRNA protein complex, and in plant cells, it's usually a perfect match with the coding sequence of some gene, okay? So it base pairs to it. And when it gets base paired to it, the presence of the microRNA and the microRNA protein complex will trigger the breakdown of the mRNA. So this is a really effective strategy to silence genes by having these microRNAs around, recognizing specific sequences in a coding mRNA, triggering degradation of that gene. The same type of thing happens in animals. It's usually not a perfect match, but when the microRNA binds to that coding sequence, it stalls the ribosome, triggers deadenylation of the 3' poly A tail. Ultimately, that leads to degradation of the mRNA. So in both cases, it's a truly effective way of silencing genes. The last thing I want to talk about is other structures of RNA molecules, and we're going to look at these in a lot more detail later in the semester. But quickly, tRNA and ribosomal RNA are absolutely critical for cell function, right? They are at the heart of translation. They are at the soul of producing proteins. And both of them are single-stranded RNA molecules. So this is a tRNA. We see several stems, several stem loops, and then right here, this is actually where the amino acid gets attached to it during the decoding process for translation. So we won't go into any more detail than that for right now, but I just want to point out that the way that it works is really cool, and it depends on some modified bases at very critical positions such that it can fold into a really unexpected structure. So here we're looking at the tertiary structure of this tRNA molecule. And because of the presence of all of these non-standard amino acids, which inhibit or prevent canonical Watson and Crick base pairing, we end up with this very, very well-defined hook or L shape, where at one end of it, we have the amino acid attachment site. At the other end of it, we have the anticodon that can ultimately recognize the codon on mRNA in the ribosome. Okay, with that, I will go ahead and end, and we will finish our discussion of nucleic acids with this.